Because it's not ants. Listen, the moment we have a synthetic ant, he's going to be yelling about it from the rooftops. Like, our neighbors are going to call concerned about why it's Balint on the roof yelling. So, when we say synthetic, I saw a question, synthetic like the Borg? Actually, I would suggest that the Borg are not particularly synthetic. I mean, that's like an interesting debate. Synthetic meaning man-made. So there's a couple different ways that we can describe synthetic DNA or synthetic animals or organisms, and they're the purists. This video talks about it a tiny bit. So it means human-made. And that can mean created a cell just from chemistry, right? We printed up some DNA and created a lipid bilayer and did all of these little things, which hasn't really actually been done to my knowledge. That's synthetic biology in its purest form. 100% of the things were created by humans. And then there's the other form of synthetic biology where aspects of it are entirely human created. Mostly when we talk about that kind of synthetic biology, we're talking about their DNA. Did we create their DNA? So you can create DNA, you can create, if the genome is small enough, so this is usually bacteria, although that's not what's been hitting the news recently. But bacterial genomes are very small and they're quite simple. And so we can actually create a bacterial genome and take out the genome. You know, there's a nice little bacteria sitting there happy and healthy in a weird scientist petri dish, and we go in and we pluck out their DNA and we inject the synthetic DNA that we just printed, and we would call that synthetic. So that's the idea here, synthetic biology, some human-made biology, human-made organisms, either just at the DNA level or just from the bottom up. And this, so is CRISPR synthetic biology, since you're doing that? It depends who you ask. It gets a little blurry. Some people say synthetic biology has to be completely like printed, constructed DNA, while other people would say yes, CRISPR, as long as you're inserting. So CRISPR can just cut stuff up and the body just pastes it back together. In that case, I would say not synthetic DNA. The CRISPR where you're actually inserting DNA, I think arguably could be synthetic biology. Although just because you've CRISPR edited something, most people wouldn't call it a synthetic organism. You need a pretty high percent of the genome.
Cass says, what about the minimum functional organism? They took bacteria, removed genes until they didn't seem to be necessary to function until it had the shortest genome possible.
Yeah, a lot of people call that, well, I'm not sure exactly which one you're talking about, but there is a synthetic bacteria that was made where they gave it like a couple hundred genes, and they found that it was like more or less functional, and they kept adding things in. And I think I've heard of the reverse where they just kept taking things out until they broke it. I think it depends on the manner in which they were taking things out, but at a certain point, yeah, it's a little bit of a gray zone. If you want to call yourself a synthetic biologist, I suppose you can, if that's what you're doing. If you're working on like CRISPR and this kind of extreme gene editing at a certain point. I mean, it's philosophical. At what point does the genome become human-made? What level of tinkering is required for us to consider it synthetic? Like if I take a bacterial genome and I've sequenced it so I know all of the letters and I just print it out and put it in a bacteria, is that synthetic? I mean, I did print it, but I didn't tinker with it in it. Balint, is that synthetic?
No, I don't even know, Lita.
He doesn't know.
You're the synthetic expert.
I'm not an expert. I'm an enthusiast. I'm a synthetic enthusiast. Star Trek question chat. Is Star Trek Synthahol made with synthetic bacteria? I love this idea. Never crossed my mind. I always just assumed it was like a synthetic alcohol, like, meaning non-alcoholic knockoff version, fancy knockoff version. But I love this idea. Or like synthetic yeast, perhaps, because like it's for fermentation, where it creates a non-alcoholic kind of, non-intoxicating kind of alcohol. I love this idea. I have no reason to suspect that that's what's happening, but I love this idea so much, Kennedy. I can't tell you. Not a Trekkie. What is is synthahol? Synthahol is this thing that people in the Federation drink when they want to be goody two-shoes and not actually consume alcohol. So you can it's like a fancy technological version of a mocktail, but it's usually beer or something like that. There someday be a synthetic Kevin. I mean, Aspergillus is not known particularly for being a key target for gene editing. I imagine that that's probably not the actual question you were asking. But I'm going to take it literally and say, one day we could have a synthetic Aspergillus fungus, fully synthetic Aspergillus fungus. That was our half of the news story. Okay, so this was 4 years ago. This was really big news on the synthetic biology front. So, a bacteria had not just a human-made genome, as in we printed the genome that we knew they already had. We pretty much rewrote the whole gosh darn thing. I mean, the scientists tried to keep the genes that were important. They didn't fully succeed, right? But the actual sequence and arrangement of these things was different than the original. So this is a pretty truly synthetic bacteria. It was still using a live bacteria, so they did this maneuver where there's a live cell, they pop out its natural DNA, its natural genome, and they put in the synthetic stuff. And they're like, it's the same thing. They do a little misdirect on the cell and convince it that nothing happened. Look, what's that over there? Is that Ewan McGregor? And then change up the genome while the cell was off. Maybe that's just me. Anyway. Okay, so that was one big advancement. Balint, would you agree that this is a synthetic bacteria at this point?
I don't know. What defines in your eyes a synthetic bacteria?
I feel like, in my opinion, this is pretty synthetic. They didn't introduce anything new. Right. But I'm not, new function. But I understand that that's, what they're trying to get to. They did that genome is completely human-made and very significant human edited. Right. Chat, is that bacteria a synthetic bacteria? Is it a synthetic organism by your own personal and as arbitrary as you want standards?
Probably.
You think so? Probably. You think yes. Chat, thoughts?
I think everyone's impressed by the pause screen.
Okay.
Sparky says yes.
Okay. All right. So there have been two big advancements in synthetic biology over the last little while. Ooh, generally we're getting a consensus. A yes consensus. Smikes, I want to know I value your opinion, my friend. Okay. So a lot of the advancements have been made in bacteria, and that's because their genomes are very small. And they're also structured very differently. They don't have there are a whole bunch of features of a bacterial genome that is just simplified compared to more complex organisms like eukaryotes. Anything that has a nucleus tends to get more complicated. And it's not just because we're dealing with more structures, it's that they're actual features of the DNA that are different and make understanding what's going on quite a bit more complex. So the goal has been for people to work towards modifying eukaryotic, so non-bacterial, eukaryotic cells. So in November, a group came out with information regarding a synthetic yeast, budding yeast, where 50% of the chromosomes were synthetic. So 6.5 of the chromosomes were very significantly edited. We can argue about whether editing counts as synthetic, but people seem to use the threshold for how much it's been edited. So 6.5 were significantly edited, and then they actually introduced a new pseudochromosome in there. They gave it an extra chromosome. They took all of the tRNA coding regions and put it in a new. So they got rid of a lot of the repetitive transposon regions. Their goal was to make the yeast genome more stable and also minimize it, start taking out the stuff that we don't need. So 50% of the yeast genome is synthetic, and and they're hoping to go 100%. So that is the best that had been done before then was like 1 chromosome, I believe.
And this is they're going in and they're just live editing in the animal, or I guess the cell?
Yes, more or less.
Like, it's not like this method.
It's not the printing method, although they did create a new chromosome also. And the method for that was a little bit different. Okay, so that was big news. And then...
Hank needs to go away. He's being distracting.
So the second piece of news on the synthetic biology front is that people have started working towards making a synthetic moss. So a plant. Their goal is to make a synthetic plant. The reason they picked moss is because moss is super cool. First of all, moss is just super cool. But also, moss can regrow from just a single cell. So it is much easier to work with moss as a plant as opposed to things like Arabidopsis or any of the other common plant models. Because they can't do that. You can't just grind up a leaf and smear it on a rock and get a whole bunch of new Arabidopsis plants. But moss, you can do that. So they thought for this purpose, it would be easier to manipulate. And the exciting thing about this is yeast, even though it's eukaryotic, so it has a nucleus, it's much more closely related to us than a bacteria is. It is still just a single-celled organism. Every now and again, yeast will clump together and kind of act like a single unit, but they're not. Every single cell is its own entity. And so the goal is to, number one, try to get synthetic plants for the same reason that they've been arguing about other synthetic organisms like bacteria. Reason it could be useful. But then also, it is bigger feat to edit the genome of something that has to coordinate between cells. We're now talking about a whole new level of complexity. And so for the first time, a group came out with a partially modified synthetic moss. It's only one arm of one chromosome 16, I believe, the small arm of chromosome 16.
Very specific.
Yeah. But it has been heavily edited. They've removed about 50% of that chromosome of things that they thought were not necessary, superfluous. And their hope is that they've recruited a bunch of other labs across the world, and they're planning every lab is supposed to take on one chromosome, and this particular moss species, I believe, has 26 chromosomes, and so they're hoping within a couple of years to have a fully synthetic moss. So this was partly a proof of principle, and also sort of a announcement of the synthetic moss program, the international program that they have established. And I thought that was pretty cool. Smikes, aren't plant genetics super complicated? Why choose a plant? I think they wanted to choose a plant because plant genetics can be very complicated. I'm not as familiar with moss genetics, but they wanted something that was multicellular. And as far as multicellular organisms, moss are less complicated than, I would honestly say, a lot of other plants. People who are really into mosses are probably going to come at me for that. But they have more flexibility and their cells are less highly specialized. And I think that's part of it. The plant cells are less highly specialized. And so I think that's part of why they're picking that. Anyway.
Is there a possibility that they're picking it for agricultural reasons downstream?
Their goal is, it's a proof of principle, but there's always this argument of we need to be able to create synthetic plants for X, Y, and Z. Mm-hmm. Can it act as an enhanced carbon sink? Or, you can throw out the ideas what could a synthetic plant do for us? Come on, guys, they already give us oxygen. What more can we ask of them?
Well, we talked about it, I think it was yesterday, of glowing plants. Lighting streets even.
Was that yesterday?
Yes, that's really cool. And is there a particular reason why, I must have missed it, why you said why moss versus an Arabidopsis or a rice or something?
Yeah, it's because those guys are stinkers to edit their seeds. And in moss, you don't, they can produce sexually, but they don't need to. So you can
so it
just
can be
edit
clonal?
one. Yeah, you can edit just one cell of the moss, and then that should be able to propagate a whole new plant.
Gotcha.
So it just makes it a lot easier for them, as opposed to every edit trying to affect a change in a seed or controlling a breeding or any of these things. Plants are like one of the few things that I've never worked on. So it all seems like a bunch of very complicated, cumbersome work that they do there. But they convinced me that moss was the proper application. Mosses are just really unique. I don't know if any of you guys are like into moss, but if you find a moss that you really like, most of them you can just kind of crush up and smear on a rock and get a whole new population to propagate. Like if you're making moss gardens and stuff, we did that in New Hampshire.
Right, your sister was telling us you can rake it. Is that the same application of you're raking it across and it's like spreading it over?
I don't remember this conversation. So.
I was just curious if that's the same as crushing it up and spreading it everywhere, as like using the rake to spread it.
I don't know.
Okay. Just curious.
Okay, so there we go. That's what we have. That's
All right.
my subject.